4 people are already viewing.
Yay! So nice.
This is good.
That sound is a terrible piece for it. Can I put it in the room? This is very terrible.
Here, take. Okay, you break the
Chan, what you need to do is put one
just
table.
put it here.
This table, I just want to put it here, like this, lengthwise, and then transfer the laptop to this table.
But there will be someone's head in front of it.
Can we just take out these two chairs? There doesn't need to be a table.
I can put it here if you want.
Okay.
It's the only
Yeah.
good solution. Nice.
Yes.
See, it's good.
Okay.
Harry,
Chan,
you
it's
all be
not.
there, right?
Yeah. Chan, it's terrible.
Hi, I'm John.
Haripriya, this is better than. It's good.
I gotta
It's
sit near
not.
the front.
Please.
So I can-
Where would you
You've
like
done
it?
so much.
There.
Let's do, yeah. Let's just take this one chair out. That's
Okay.
all. This is the only chair that we need to shift out.
Also,
Yes,
Harry,
yes.
just to get on that, that'd be fun.
Thank you.
I gotta be gone by 5, so don't run over. And
That's
anyhow,
excellent.
the university requires that the examination be finished within 3 hours.
Yes.
And I hope it is. For you, people within 2.
I hope so too. Because then that means we'll have 1 hour to party outside there.
Say again?
We'll have 1 hour to party outside there.
Yes.
So we have to run away at 5, then you will get 1 hour with us before you leave.
Right.
That would be
So
awesome.
don't overrun.
Yes.
That's what I'm really getting at.
Thank you.
This is better.
Yeah. So can you move this? See, this is nice.
Good.
Yeah, this is really nice.
Happy?
Yeah.
Awesome.
The slides are visible, right?
Yeah.
Did the link work, Hari?
Yeah, I just
Awesome.
it. Should I just leave it as a separate?
No, you can just be it in the same deck because that way you don't have to do anything and it will all transition. As long as you're not connected. Oh, that's perfect. Actually, you can leave that as a first title slide. They're actually in.
You got a jacket on? I got a jacket on. Good for you. Yeah.
Take some pictures, maybe.
Well, I gotta be gone probably at 5, so I tell if I that's overrun.
5 is plenty time.
Yeah.
I want to hear about your history because we got some emails.
Archana is already saying congratulations.
Which you were not CC'd on, with the idea of making sure that we can remind the questionnaire things that should be asked.
Can I change?
Oh no, it's just the other way around. You have to rotate it.
Okay.
But I don't recall that you had.
Just take it out.
No, I never wrote back, but some people in that list wrote back
Yes, I did.
talking about, you know,
Most
many
of
things
them
I
had-
didn't know about.
Most of them had how cheap I was. Yes. I said I prefer the word frugal, which is under you.
Yes. No, I would much rather use the letter. No. I think I think the point was that it's just that the person who is doing the interview might not have known the historical aspects.
Well, why should he? No, actually, I've known Andy for a long time, because he used to run the Stanford Instructional TV Network.
Well, I do. I used to have a TV network. Wow. So it would be the for the space.
Yes, well, before the internet.
Not just, yes.
They were broadcast live to those companies.
What?
Yes.
I
You had a sound box.
I'm going out,
Yes.
getting people.
Well, you had a
But
classroom
this,
too.
this is.
Yeah, yeah, yeah. Like whether or not. It has
Yeah.
one
So-
camera to look at you, one camera to look over your shoulder, and one from the students to sleep.
So all the things that they talk about is moving and stuff.
Yeah.
You have a broadcast.
Yeah. But what they found was really good thing. Was like close to video. Look over.
The thing is not switching on. I don't know what's up with these. I made it into a setup where it won't matter. So
I see.
forget it. No, don't connect. This is terrible.
I'm in the genetics.
I sent you 3 emails.
I think you're press right about you.
Here we are.
Okay, good. I'll come and talk to you.
Yeah, absolutely.
You're not
Your
staying?
team is
No,
not
I haven't
working.
spoken then.
Yeah, so,
Did you put it in before or after?
Yeah, I didn't know about the moon. So can I take it out? That's really nice.
well, I mean, that was a long time ago. It was before it was 3 years before like, usually just send out.
No, I bet.
Do you think there was a call?
Well, what happened, I was insulted a bit for Hughes.
So 3M.
Hughes had just
And they're like
landed the
dental?
surveyor satellite.
I
And he
see.
put it on camera.
Okay, we're good.
Taking pictures.
In their,
It was brilliant.
Yeah.
Incredible. And it all the way up. It's 300 meters.
Oh, sure. No, this-
yeah,
You
so.
won't
So, so that was my idea. 3 years later, pick it up and bring it back, so.
Yeah, but you can't do like the lower, for example. or-
So what many counts have
I know.
But this mom.
Yeah, and you can just like sit on some teeth together.
Get the salt, boundaries.
Yes.
I'm Sonia.
It's nice to meet you. Yeah. It's good to finally meet in person.
Yeah.
No, I don't think.
That's fantastic. Yeah. I hope you also have some coffee.
Oh,
and stuff
yeah.
outside too. Excellent. Thank you. Nice to meet you.
It's working. Down is down.
The more I thought about that,
You're
the
after
world,
the university.
meeting is smart.
Yeah.
Have fun. Enjoy yourself. You rock the party. You know that. Take care of everything.
Your
charger?
Your thanks, yeah. It's gonna work its way out.
No,
Yeah,
so
me
leave
too.
it here. There's something
Like,
wrong with
hard
this.
to- right, no, it's just one of those.
Yeah. It will not,
There's
nothing much.
But the thing
I
that I'm
am.
really hard about is when I focus on
Yeah.
the
That's a very good idea. I'm also going to put the minimum stage right now.
And so basically, half my time is, I mean, regardless, like, the story is really exciting, but I need to get it out.
Charged in yes.
Yeah, this is also fully charged.
Yeah.
Half and half.
Yeah.
Yes.
It's a very big, really nice package. So like being with faculty, going through their plans.
Oh, good.
Finally.
Alright, how can we reframe this so that like, oh, you're
It's
fine. Carolina.
Well, it's actually.
So part of me is kind of good. So that was like, okay, I go here at Stanford, and what I do here for free. So
I like meeting convention tools like
but yeah,
there's a lot there's like
basically they say in the fashion
it's like thanks a lot. Like double
sounds dope. Yeah. But just in like, yeah, it's like not gonna stay. I think the thing that's hard is that it's like things are really cool, but it doesn't move you forward on the track, and it doesn't move you forward on the track. Well, why wouldn't that? Why wouldn't it?
I think
Like, because I'm going to be publishing.
So excited by it.
That's true this 1 year. Well, you just write all my Also,
Yes. It's really nice to see you. Yeah, thank you for coming. Thank you. It's so great that you can make it. I'm also looking forward to seeing your grandson.
I think it's fun.
So yeah, we actually saved this place for you all.
You're on fire.
Hey.
Yeah.
Or here, some flyers. I put out as many as you can. Also,
I was telling Laura that I was sorry. I was last for myself and I was like on the train right when I was having his twins. And they're like 3 years. Yeah. Yeah. It's been I literally was like on the street. Yeah, I know, I was super excited. So like,
I think that's mine. Mother, that's mine.
That is yours. I wanted to just make sure that you have all the one person who had to actually last minute.
Hi, nice to see you.
Nice to see you. I was so wonderful. I'm excited for this. Yeah. Are we still in the front?
Yes, so we make these chairs for you.
Someone's all set.
Yeah, this is great.
Where's this sit right here? Okay.
Because
Bring me from clothes on.
you cannot have leaders or anybody
Oh,
outside
yes.
part of
Oh, no, I was just late, so.
I'm volunteering, and everybody else is the leader for the business.
Yes.
I didn't find.
Yeah. So I do want to read it.
Yes, you are, yes.
Oh, this is a pickle for this next.
Yeah. Let me know when you guys are. Just wait for everyone.
I think I'm gonna use that as like making free and making different. Like climbing out. Yeah.
It's more about the position.
Yeah, it's like. Yeah, so it's.
So they have to be. You can rearrange that.
Well,
Birds on that. You'll introduce it, I guess. Yeah. I mean, you have the first, the second.
That's correct. That's correct. Yes.
Like,
Thank you everyone for coming. This is Haripriya Mukundarajan's PhD defense in the Department of Mechanical Engineering. I'm assuming we have all the committee members in the room, so we can actually officially begin. So I think one thing that I want to say before we start is let's figure out how this works. That or that? Okay, I'll just do this here. Oh, there is a antivirus protection expired. Or I'll hit okay. Okay, I'm sorry. Okay, you're in for a treat. I think one of the aspects of when Hari joined my lab, it was the very early days. She was one of the early pioneers that built the lab. And you're going to see a very broad range of things that Hari has done in the lab and how her research has taken flight in many different directions. And I think I want to highlight one aspect of this that you would see when you start asking about the problems that we face in the world, going beyond our academic circles, the kinds of health problems, the kind of environmental challenges that we face on the planet, you really have to think about planetary scale measurement tools. And I think one hint in Hari's work that she will talk quite a lot about is the solutions that she's been working on have the capacity to truly scale to that planetary scale. I'm incredibly excited about the fact that these sets of solutions have deep insights, but at that same time, we might be able to actually implement them in the real world. One thing that happens when you go on in a space of doing a very broad PhD is with a student, their character shines as you go along. I've had plenty of time that I've spent with Hari in the lab. And this picture sort of depicts a little bit of that character. She is literally building a model of a mosquito. And so there are several things I want to highlight here. First of all, her character, she's doing this for somebody else. And so when she dedicates herself, many times I've seen this in her work, where she's very broadly thinking about other people. And you will see that over and over again in the kind of work she's done. But of course, she brings precision to even the smallest task. So of course, there has to be a ruler and a calculator involved in this process of building a mosquito. I've had the chance to go out to the field with Hari, which is another part of this work, that we've taken some of this work out to the field to test it. And the picture, on the other hand, highlights us in the field. And I want to point out something very particular, which is very difficult to notice unless I point it out. There are multiple different nationalities represented in this picture. This is Erica. She has roots in Mexico. She's very well dressed up, hoodie covered, shoes. This is Amalia. She's from Greece. Amalia is very careful. She even has a head cover so no mosquitoes can bite her face. Here is a Dutch postdoc, tall and built, and look at all the socks, no areas covered. And here is Hari, right there, open toes, sandals. We're out in the field. And ironically, because of that, and of course, because sometimes in the early days, they used to listen to their advisor, and we are standing literally 1 feet away from a massive poison oak tree. And of course, we were searching for mosquitoes, and I championed not realizing what poison oak actually is. And of course, with the international character of this lab, none of us knew, and we dived into hunting for mosquitoes, and the rest is history. I'm amazed that she didn't say that I quit, and this is not, that would have been my fault. But she championed, and we actually realized some very interesting things about the local ecology here. But most importantly, not to do this out in the field. And now I always tell people how to identify poison oak. I think, in all seriousness, Hari brings a kind of a character to the lab that I've seen very rarely, where if she is excited and charged about something, she will execute it. She was the first person in the lab, and actually we were thrilled at that time because when she joined the lab, I was excited about mosquitoes, but there was nobody on campus either working on mosquitoes, growing mosquitoes, and I talked to people and colleagues, and there were no mosquito facilities, and so Hari literally built the first one. And in the first 2 weeks of her here, we were rearing our first sets of mosquitoes. She takes a lot of the work that she's done in the lab out to both inspire people, and this is something that I'm very proud of, because in the end, much of this matters when it gets shared. And we've had a fantastic amount of time, Hari, so thank you so much for what you've contributed to the lab, to the science. And I think one word that I often describe Hari as is she brings both the fun and the mentals to fundamentals. Up for some fun.
Thank you for that fantastic intro, Manu. And thank you so much, everybody, for coming to my defense today. It's great to see the room so crowded, because I think today is the only day of my life that I'm going to feel almost as popular as Eddie Redmayne, right? So my story today is about 3 fascinating creatures out here that are going to highlight the role of dynamical phenomena, nonlinearity, emergent behavior, and collective dynamics in shaping the ecology and evolution of biological systems at various scales of organization. So first, looking within an organism itself, I'm going to tell you very briefly about my work on sequencing the transcriptome of placozoans to study their biology. Next, I want to show you an unusual flight phenomenon in water lily beetles to highlight how dynamical interactions with the surrounding environment can shape the ecological niche of an individual organism. And finally, I'll describe a way to get my slides back first.
I think it's in the advancer that it goes.
Probably, yeah. So finally, I'm going to talk about a way to look at dynamics at the population level as to how we can track entire populations of mosquitoes and study complex interactions between species with the goal of elucidating disease transmission dynamics and complex host-vector pathogen interactions. So let's start our journey at the very bottom of the ocean, where animal life first evolved with the placozoan. So the placozoan is one of the most early diverging metazoans, and what you see in this video here, the white shape-shifting blobs, are individual organisms at the bottom of a culture dish filled with seawater, adhering to the glass surface and feeding on algal mats. So these animals have the fewest known cell types of any metazoan, arranged in a simple structure of a flat 3-layer sandwich. The placozoan also has the smallest genome of all animals, with only 100 million base pairs organized into about 12,000 genes. So I worked on sequencing the transcriptome of placozoans, as very little is really known about the expression of their genes and their physiology under various conditions. So I picked up animals from various phenotypes in culture and prepared libraries. And with the help of Sophia Ruiz, a postdoc at the Roxar lab at UC Berkeley, we assembled a high-quality transcriptome where we identified about 12,400 genes and annotated the genome more precisely using our transcriptome. And we used an in-house pipeline developed at the Joint Genome Institute to do this. I focused a lot more on the latter half of the pipeline, looking at differential gene expression between these phenotypes that we see in culture using these packages, Callisto and Sleuth, respectively, to estimate transcript abundances and test hypotheses for differential expression. So the insights that serve as supplements to biophysical observations in the lab, I'm going to briefly outline two examples of this in this story. The first one is when we take animals and make wounds in them and allow the animal to stick itself back. What is absolutely astounding is that there is no transcriptional signature that we observe for either tissue damage or repair in this animal. If you look at the plot here, each dot on the plot represents one gene. And the x-axis shows you the average expression of that transcript in an animal under normal conditions. And on the y-axis, you see the expression under wounded conditions 4 hours after making the cut. And what is incredible is that all the dots fall along the diagonal line, which shows that there is statistically no significant difference in expression between normal and wounded animals. This is actually consistent with another project done by a postdoc in the lab, which shows that the extreme shape change that you saw in the first video actually arises from microfractures, where the animal slightly breaks itself and sticks itself back, showing that this is a physiologically normal thing for the animal to do, with cell-cell adhesion having fundamentally mechanical origins. Another really interesting insight was with animals that detach from the bottom and float all the way up to the air-water interface. So I prepared these plots where I functionally categorized all the transcripts in the animal and estimated within each category how many of the genes were either upregulated or downregulated. And what this plot here told me was that animals that float up to the surface have much reduced DNA replication, very little cell proliferation, a steep reduction in all kinds of metabolic activities, and reduced ribosome production and protein translation. So these are essentially animals that are sort of in a state of stasis. On the flip side, though, they do upregulate plenty of elements that are associated with signal transduction between cells. This is a pretty interesting observation that probably has a lot of implications for ecological transitions, where the movement from the bottom of the ocean to the interface could have implications for the animal's life cycle. But at this point, I'm going to ask all of you now to make the same transition along with me. From the bottom of the ocean to the interface between air and water, and look at another evolutionary and ecological puzzle in insects this time. Through this incredible flight mode called interfacial flight in water lily beetles. So insects are the most evolutionarily successful terrestrial animals with the ability to conquer all kinds of niches on air in ponds and different arboreal environments and so on. And this incredible diversity of species that we see in insects is fundamentally thanks to their power of flight. So there are two competing stories for how insects first learned to fly. The first is the arboreal hypothesis of flight evolution, which is very nicely illustrated by these gliding ants, Cephalotes atratus, which live on the trunks of extremely tall trees in the tropics. So what you're going to see now is a video taken from the top of an about 100-meter tall tree, where first there's going to be a styrofoam ball that's dropped and it falls straight down. And now an ant is dropped from the same place, and you can see it flies back to the tree. And it actually does this by moving its legs and abdomen in a way that allows it to shape its trajectory and glide back to any object that provides the white contrast of the tree trunk against a dark backdrop. The other hypothesis for how insects learn to fly came from looking at the surface of water with the premise that ancestral crustaceans had gills that they flapped to propel themselves along the surface of water, like this creature here. And as these gills grew stronger and more specialized for propulsion, they eventually became homologous organs that are basically the wings of modern insects. So this hypothesis was fundamentally based upon a series of observations in stoneflies, which are the most evolutionarily basal of insects. And this is what you see in stoneflies. You get some animals that swim along the surface of water, and some others which swim and skim using both their legs and wings. And then you have those that basically use their wings as paddles, followed by something that you can actually call flight. But in this case, the animal is flat, holding onto the water with all its legs and flapping its wings in the air for propulsion. Slowly, they get the confidence to get rid of the middle training wheel legs, and eventually, you have another progression where they're just on the hind legs. And this leads to a progression into airborne flight. So this was the amazing aquatic hypothesis of flight evolution, which presents a very fascinating perspective of one possible route in which insects could have first learned how to explore the aerial environments and conquer the world. And there were certain advantages to thinking about flight in this way. For one, weight of the insect is supported by surface tension. And so you don't need to develop strong wings that generate sufficient lift to hold its weight aloft. Primitive weaker wings are totally okay to swim smoothly along a water surface. And as the wings grew stronger, the theory went, there would be a smooth transition from water to air, overcoming the very low resistance found on a water surface. But the question here is there really such smooth sailing on a water surface? And that's something you can only tell by performing a quantitative physical analysis of the flight mode in these insects. And that's where the star of this story comes in. This is a water lily beetle, Gallericella nymphaea, and Manu found this beetle when hiking in Harwood Forest, zipping between water lily leaves on the surface of ponds that it basically munches on for food. So take a look at this video that illustrates this phenomenon with 4 legs on water and interfacial flight. But that's not the only trick that this insect can do. This amazing video shows the insect starting with unfurling its wings and beginning to execute interfacial flight on 4 legs, and then moving to 2 legs, going up into the air, and it's even capable of adjusting the plane of its wings to hover and fly backwards as well. And this incredible number of transitions from the interface up to the air makes it an amazing model system for analyzing the biomechanics underlying the aquatic evolution hypothesis.
Did you make that video?
Yes.
Fantastic.
So the adaptation that allows this insect to fly on the interface comes at the joint between its legs and its claw. The legs are covered in hair and repel water, and the claws are extremely smooth and have a propensity to be wetted by water. And this sharp transition between the hydrophilic claw and the hydrophobic legs is right at the joint between the claw and the tarsus. And what it implies physically is that the water meniscus has its contact line pinned to this joint right along this interface. And what does this mean for the insect? It means that when you pull the leg up, as you see in this photograph here, the meniscus goes upwards, and surface tension consequently pulls the insect back down, preventing it from popping off the surface. And conversely, when the meniscus is bent downwards, surface tension now provides weight support for the insect, pushing it back up. We analyzed the kinematics of interfacial flight using high-speed video recordings, and an automated script developed by a high school intern, Aditya, was used to track natural markers of high contrast on the legs close to the point of contact with the interface. So we saw an oscillatory trajectory, which is something that you would expect for an insect with a periodic wing beat propulsion. But what is actually interesting was the upward velocity of these vertical oscillations leads the power stroke of the wing beat, indicating that there's some kind of complex energy storage and release in every wing beat cycle. Using these videos, we were also able to compute non-dimensional parameters that give us the ratio between different forces and their relative importance in interfacial flight. And so we were able to conclude that inertia is the dominant effect in this mode, with significant interfacial contributions and negligible viscous drag. We used these insights to go ahead and build a dynamic model where we wrote down all the forces acting upon the insect in interfacial flight. And having done this, look at this incredibly complex model with a lot of joints and many different shapes. We decided to go with a first order approximation where we reduce the insect to a point mass pinned on the interface with all the forces acting right at this single point. So how do we start writing down the equations? We start with airborne flight because that's something well known and well studied in the literature. In the horizontal direction, you have thrust from the wings, counteracted by air drag. And in the vertical direction, the wings provide lift with which they have to overcome gravity. Next, we add the forces that come from the contact with water. The straightforward ones are just drag from water. And the interesting ones are from contact with the interface, capillary wave drag in the horizontal direction and surface tension in the vertical direction. To provide a detailed model for each of these forces, the wing force is fairly straightforward. It's simply a sinusoidal forcing function which we measure from the videos. How do we estimate the magnitude of the wing force? That's a little more challenging, and that's where we use the help of videos like this one, where you can see the insect is directing all its wing force into lift, but it remains trapped on the surface. So that gives us an estimate that the maximum lift produced by the wings, so the maximum force produced by them, is probably less than the sum of its body weight and surface tension on two legs. And that allows us to put a bound on the force generated by the wings. The drags are fairly straightforward. The insect is a bluff body moving through a liquid in cross flow, and we can write down the normal equations for viscous drag. But capillary wave drag is a really interesting term that crops up here. I want you to take a look at that video and look at the optical post and how you can see ripples deforming the shape of that post. These ripples, which are shown in high contrast in the photo here, are what generates this phenomenon of capillary wave drag. When an object is traveling on the surface of water, the ripples move ahead of the object. So long as the object is slower than its ripples, no problem, everything's fine. But the moment the object starts traveling faster than the ripples and starts cutting through the ripples that it produces, it essentially experiences the interfacial equivalent of a sonic boom. And these ripples now start dissipating energy away from the insect, which can be manifested as the capillary wave drag force proposed by Raphael and Dejean in around 96. Since our beetle is extremely small with tiny legs and inertia dominates surface tension, we are in a regime where we can directly use a dipolar approximation model for describing this force. And then we move on to surface tension, the other really fascinating interfacial force. As I showed you before in an earlier slide, we use the fact that the meniscus deforms and is pinned at the legs to compute this. The meniscus forms a minimal energy surface, so all we have to do is balance pressures on either side, and that gives us an expression for surface tension based on the shape of the meniscus. The obvious thing you can see is that the meniscus is not a straight line. It doesn't form a triangle. It forms a curve with that funny shape out there, which gives rise to this extremely nonlinear term in the expression for surface tension, making this a force-damped nonlinear oscillator. So we use this model to simulate some trajectories for this insect, and the simulated trends closely match what we measure from the experimental kinematics in terms of the oscillatory profile as well as the general velocities that we see. And the test that we really use to verify that our model is right is that when you change the angles, thereby changing the ratio between lift and thrust produced by the wings, you should have a transition from interfacial flight to take off and go into air. And that is exactly what we see when we produce trajectories at different lift thrust ratios. So now we can start asking the really interesting question. How easy is interfacial flight? Easy enough for weak insects to do it and take off into air? I'm not sure what exactly I did here, but I hope you'll give me a moment to make this up. This is probably a bit of a challenging situation for my laptop to handle. So yeah, maybe it should consider going back in the interface. All right, here we are. So the first thing that kicks in as you start moving faster and faster on the surface is, remember, I told you, you're going to cut across your own ripples and hit capillary wave drag. And capillary wave drag increases as the sixth power of velocity once you cross that critical threshold, which means an insect slower than its ripples because of its weak wings would be totally fine on water. But the moment the wings start getting stronger, the capillary wave drag introduces a huge energy dissipation on the insect that has to be compensated for by becoming even stronger. And this actually makes it even more challenging than airborne flight at the same speeds. The other thing that we see is that the trajectories become aperiodic as weight to lift ratios increase. This plot is basically a stroke of the trajectory of the insect. What it means is that we measure the vertical displacement periodically with a period equal to the wing beat. So if the trajectory were perfectly periodic, we should get the same displacement in every single cycle. But for a completely aperiodic trajectory, it could be anywhere vertically at the same phase, which means what you really get on this plot is a smear, and that's exactly what you see in that mid-region that corresponds to approximately the same wing strengths that you would need to actually support your body weight and take off into air. We also get self-intersecting trajectories in a phase space of displacement against velocity. And what is really interesting to compare here is with other known chaotic oscillators in surface tension-based systems, like this one, which is a droplet of water bouncing upon a soap bubble film. Even though they're both phenomena driven by surface tension, the equations are slightly different in the two cases. And yet, the nature of the phase space trajectories that we get for both these problems are highly similar to each other, providing another corroboration that this system is also a surface tension-driven chaotic oscillator. The trajectories, when simulated, indeed have sensitive dependence on initial conditions, the classic hallmark of chaos. And when we go back to experimental data, a delay plot shows absolutely no correlation between the displacement from one time step to the next time step, which is a very good measure of experimental chaos. We also simulated for different frequencies and different lifts, what would the takeoff times of these insects look like? And you can see there is a wide range of takeoff times, including some values that are too fast to be under active neural responses, which means that there's probably no active control in some of the trajectories of this insect, which you might see in these spectacular fails that it demonstrates from time to time. So let's all take a moment to laugh at how clumsy this guy can be. I showed you this one earlier where it's incapable of braking once it hits the edge. And also this video where the legs just get stuck on the interface and it can't actually take off when it wants to because the legs get wetted. And how robust is a situation where whether you take off or not, which can determine whether you get eaten by a predator or not, how robust is your flight mechanism when you have no control in several situations over whether or not you can escape? So that brings us to a conclusion for this. It shows that although we initially thought that sailing smoothly on a water interface and taking off once our wings became strong enough might be possible, flying on a fluid interface is actually an extremely bumpy ride, and it may be extremely challenging to evolve active flight control in an unpredictable environment of this nature. So at this point, I'd also like to thank my co-authors on this project, Thibaut, who did a lot of the experiments and collected many of the videos that you saw, Donald, who just started the experiment even before I joined the lab, and Aditya, the amazing high school student who, even when he was an 11th grader, wrote the automated script to process our videos and extract all the trajectories. I also want to thank David Lentink and Paul Mittigi for all their inputs on this project, which went a long way in shaping how we approached this problem and corrected a lot of the pitfalls that we had in our analysis. What I really got out of working on this was firstly a taste of biomechanics and working on organismal biophysics, but also an appreciation for the role of nonlinearity in biological systems. And this was the first time that I realized the kind of intersection there is between dynamical system studies and various areas of ecology and evolutionary biology. And this is the kind of idea that I want to transfer now from the individual scale to the population scale in a different insect, the mosquito, where I'm going to spend a little time talking to you about developing tools to generate data for vector pathogen ecology. So I'm primarily going to discuss this project that we called ABUZZ, which is crowdsourcing mosquito surveillance using mobile phones to record their sound. And this work was also published a couple of years ago in eLife with my amazing co-authors, who I'd like to thank again, Felix, Erica, and Cooper. I hope all three of you are listening. So if you think about the world that we live in today, it's pretty, how many of you really found where this room was by pulling out your mobile phone and trying to get here? Well, there are certainly some of you. So it's natural to expect today that I can just get my phone out and expect it to give me some kind of information about the real world around me. So that's why I think it should be totally reasonable that I pull my phone out, and when I go, to some nice national park to celebrate this weekend, am I going to get bitten by mosquitoes in this spring haze out there? Like, why doesn't my phone tell me this useful stuff? And it's not just applications like, am I going to get bitten or sick while camping? There is so much powerful insight that we can derive from data of this sort from a huge data set. The first one would be identifying spatial hotspots of mosquito abundance, which are also usually associated with hotspots of disease transmission and usually require targeted control to prevent disease outbreaks. The other aspect is temporal. I want you to look at this graph here, where the blue bars represent the number of dengue vector mosquitoes in a given island, and the red line shows the clinical incidence of dengue fever on the same island. And what you need to notice here is that the peak abundance of mosquitoes leads the peak incidence of disease by a period of about 4 weeks, which is actually sufficient time to go in and perform an intervention that might have even prevented that outbreak. And this kind of insight, tracking the population, can be incredibly useful in extremely accurate short-term forecasting of disease outbreaks. The third amazing application we can have with such data is personalized vector control and disease management in this era of personalized everything. Take, for example, this picture of a mosquito. If you were sitting in a village in Africa and you got bitten by this mosquito, how would you know how to control it? It could be any of 3 species that are visually indistinguishable, with the caveat that one of them would bite you inside the house, so you could use bed nets. The second, however, would get you in your garden, so you couldn't use bed nets. The third doesn't even care about you, it just wants to go for your cow, so you don't have to worry about it at all. So knowing only that you had that insect that you trapped, how could you decide exactly what kind of vector control approach to apply in this situation without performing expensive molecular assays? And this is why we need this holy grail out here. Global maps of real-time species-specific abundances of mosquitoes at extremely high resolution across the world. And the fundamental requirement for making maps like this is lots and lots of data from the field. Although the reality that we have today is fairly sparse inputs of field data in mapping of mosquitoes. In this map, the black dots represent field observations, while all the color is mathematical modeling that takes these field observations and extrapolates it across the entire continent. And as you can see in this region here, there's a huge swath of the continent where we expect extremely high densities of this disease vector with only one verified, standardized, reported observation right out here. So why can't we do better? I mean, is this like inherently hard, or is there something we're just missing out? It turns out that this really is an extremely challenging problem. You need a lot of time, effort, equipment, labor, skilled personnel, and so on to carry out any of these activities to record mosquito abundances out in the field. To lay out traps, to process the mosquitoes from these traps, to identify their species under a microscope, to test them for infection. This is an extremely labor-intensive process that could certainly do with a lot of automation. And if you thought the logistics were tough normally, they get even more challenging in field sites like our field site in Madagascar here, where, just try lugging your battery pack up that hill and see how many days you want to do it. So what this fundamentally means for the problem is that while we'd like to look at mosquito-borne disease ecology as a big data problem, we're actually stuck dealing with a very big problem of having far too little data to work with. So our group works on 2 parallel methods for generating this kind of data. One of them is on the side of molecular surveillance, based on the idea that mosquito saliva, when a mosquito bites you, contains both the pathogens as well as a little snippet of the mosquito's DNA itself, and you could exploit this to create microfluidic devices that are essentially skin mimics that induce the mosquitoes to bite, where they obligingly deposit these samples that can then be processed in high throughput. So I spent a little time thinking about this project, and it's now being led further by 2 amazing postdocs, Felix and Shanna, who've achieved some pretty incredible work that you can see out here, getting the mosquito to actually bite that little spot. But the project that I'm going to talk to you about is on the side of surveillance of mosquito species rather than pathogens. And this is based upon a very simple concept, that mosquito sound is very useful for tracking the species. That's because this is the kind of insect where when they mate, the male has to really listen very, very carefully to the female and get onto the same exact frequency such that the second harmonic of the male matches the third harmonic of the female, and only then the female is going to give the male the time of the day. So what this means for us is that mosquito frequencies get self-selected into very species-specific bands that we can then use as species identifiers. This insight has been around since the 1950s and has even been exploited in a couple of traps that play the sound of females to lure the males in and kill them. But why can't we use this for surveillance? Here's the challenge. You want to use equipment that's sensitive enough to record these faint buzzing sounds, but also cancel out everything in the environment around, You want it to be rugged enough to work in all kinds of conditions, portable so that you can put it up on a tree like this, and cheap enough so that you can put it in everyone's grandmom's backyard, right? So that's not a lot we're asking out of this equipment, but it so happens that this is a solved problem these days. Each one of you is actually carrying around this amazing acoustic detector right in your pockets out here, which just happens to satisfy all of these conditions precisely. So, yay. And that gives us the solution of using mobile phones to record mosquito sound for citizen science-based mosquito mapping. This is how it works. Consider any place in the world where you want to sample mosquitoes. First, you need to know how the mosquitoes there actually sound. So you go out and do a baseline survey where you record the different mosquitoes and prepare a database, which you can then use for reference. When people out there in that place record data and send it in using different methods by just pointing their phone at the mosquito, the phone collects both the time and the location metadata as well as the sound file. So we then take the sound file and pull out the frequencies at every instant of time and use this frequency distribution to match it against the database that we created and identify the best match and the most likely species. This is now paired back with the time and location metadata to put a dot on the map. And the more dots that we create, this is how we get closer to our goal of high-resolution mosquito maps. So I'll go into a little more detail about each step in this process. The first is recording mosquitoes, which you can do, as you see, either in free flight or in confinement by capturing them in cups or in bags or in cages and so on. And this is how the mosquito sounds. So that is a recording that I collected of the Anopheles gambiae malaria vector in Africa using my personal mobile phone. So we take that raw audio signal and convert it into a spectrogram, which basically pulls out the dominant frequencies in the sound at every instant of time. And we also have a script that pulls out the fundamental frequency, the lowest value, which corresponds to the frequency of the wing beat of the mosquito over all these instances. We take this wing beat frequency and rather than averaging it, as a lot of approaches do, instead, we construct a distribution of how often we see a certain value of wing beat frequency produced in that recording. And we use that distribution for trace matching to identify the species by comparison with our database. So the first question is, if frequency is so important, how do I know my phone is recording it correctly? And to verify that, we performed an independent test where we had a high-speed camera recording a tethered mosquito at 10,000 frames per second, synchronized with the phone recording audio from the same mosquito, and both of these traces were aligned using a buzzer and an LED driven using the same pin of an Arduino to produce the same signal code. And when we do the alignment, we find that there is an exact correspondence to within 2% of the frequency, which is basically the computational accuracy of our algorithms, which means that yes, phones do capture precise wing beat frequencies. But do they do it as well as the fancy microphone that I showed you earlier? The answer turns out to be surprisingly, yes, they do. The gray band on this plot here, between the black and the gray lines, represents the range of performance of standard industrial and studio microphones that are out there on the market today. But if you look at these colored traces that correspond to different mobile phone brands, including this red one that corresponds to a $20 flip phone, all of them lie pretty much within that gray band over a wide range of distance, which means that yes, your phones are actually as good as some of the best studio microphones out there. This experiment also gives us a range for how far you have to be from a mosquito to record it. The point where the signal-to-noise ratio basically dies out gives you the distance, which even for the flip phone corresponds to about a couple of inches away from the mosquito that we anecdotally hear and from experience do know that it's actually pretty easy to achieve this kind of distance out in the field. So yeah, at this point, I mean, this has been a lot of work, and it's taken me a long time to, stay here in the middle of Silicon Valley and do all of this stuff. But even after 7 years in Palo Alto, I believe in a world beyond iPhones. So that's why it was important to us that for this solution to be truly scalable, it has to work not just on this fancy smartphone out here, but also on the dumbest of dumb phones, like this one that Erica produced as something she used in her middle school days. So we used these wide range of mobile phones to record the same cage of mosquitoes, and the frequency distributions that we measure are statistically indistinguishable from each other, implying that any piece of data that anyone on Earth collects using any kind of mobile phone is quantitatively comparable using the same exact analysis pipeline, which is absolutely fundamental for a global solution to this problem. So the phones are great, but does biology cooperate? Do species actually sound different enough for us to go ahead and do this? So obviously the first step is to figure out how species sound, and there was very limited data available on this when we started working on the problem. So we spent a lot of time at various labs, including the CDC, who were instrumental in helping us create this database from 20 different species to survey the wing beat frequencies based on recordings from caged populations. So the question you can obviously ask me is, did I sample these well enough? Are they representative of that species? The answer to question 1, yes, I sampled them pretty well actually. And the way we tested that was to draw subsamples from each of the distributions. If I pull out, say, 10% of the total number of observations in a distribution, and now I compare the two distributions, if I find that the divergence between them is extremely low, that means the two distributions are very close to each other. So it turns out that even at around 10 or 20% of a certain sample, the distributions basically converge, which means that they're pretty well oversampled. The other question is, were they representative of the species? We did this by testing many different strains for the same species. In the case of Anopheles gambi, the malaria vector, the 3 strains there are the wild type, a strain that's resistant to the insecticide permethrin, and another strain that's resistant to bendiocar, another insecticide. And these 3 sound pretty much the same. Aedes aegypti, one of the common threats worldwide, was another case study where we had colonies sampled from 3 different labs in 3 different physical locations, and those sound pretty much the same as well. We did find an exception with this species where a strain from Sudan sounded extremely different from a strain from Senegal. We don't know exactly what it was, but when we got back to them and asked, well, they might not have been the same species to begin with, so maybe we're witnessing an interesting ecological event of species divergence out here. So there are some species that are highly distinguishable based on data from phones. For example, Anopheles gambi, the malaria vectors, are very easily distinguished from Culex pipiens, which are very common but a lot less harmful than these ones. The same is true for the Culex pipiens and Aedes aegypti, the main Zika vector in Brazil. There are also situations where entomologists out in the field would like to distinguish closely related species that look very similar. In case of Culex pipiens and its cousin that stay out here in California, or these 2 Aedes species. And while you need a microscope to distinguish them normally based on their morphological features, you can do it just by pulling out your mobile phone, recording them, and seeing whether or not they have a higher or lower frequency. And you remember the mosquito I showed you there that had 3 indistinguishable subspecies? It turns out that all 3 sound pretty divergent from each other, so you would not have to do a molecular test to distinguish them. All you have to do is take a phone recording. So how do we identify the species? We do this using an algorithm called maximum likelihood estimation. But I told you about a refresher. For the spectrogram, where we pull out the frequencies and create the distribution. Now we take that distribution and compare it to the master data set that we created and score every species based on how likely it was that this recording was subsampled from that master data set. We then pick the species with the highest score and assign that as a species ID, and this also gives us a confidence probability for the identification. So when we test this against data collected using the same phone, it's pretty good. If you look at the predicted species in the vertical direction, the prediction corresponds pretty well with the actual species with everything falling along the diagonal, and that's awesome. But the moment we start mixing data from different phones, even though it should all work, like I said earlier, we get this hodgepodge where the algorithm starts making a lot of mistakes. And that's not because the algorithm is inherently bad. That's because this is a really, really difficult problem to solve. You have 20 different species with fairly wide distributions and significant overlaps between the distributions, which means taking one particular sample and trying to tell which of two closely spaced distributions it belongs to is inherently extremely challenging. But that's where the power of using a mobile phone comes in, because the mobile phone also gives you metadata for time and location. Take this example between the Aedes aegypti Zika vector and the Anopheles gambiae malaria vector. They sound identical, but one of them is going to bite you during the day and the other one's going to bite you at night. So depending on when you got bitten, you can see what you have to worry about when you go to the doctor. These anophelines are another example. They sound identical, but live on completely different continents, so you don't even have to worry about comparing them to begin with. So based on this, we took open source data collected by the US Army's Walter Reed Biosystematics Unit and prepared a location filter for our 20 species for 200 different countries. And when we applied that location filter to our estimation algorithm, the classification accuracy doubles from 35% to 68%, with a lot of recordings falling back on the diagonals again.
Does that include time as well as location?
So we haven't included time yet. That's a work in progress because we're collecting more data on circadian rhythms, as I'll come to soon. All right, so 78 is great, but I'm still going to get a C grade if I got 78 on a class, right? Can we do better? We've been trying to push the envelope for accuracy using supervised learning algorithms with the help of Sam and Abu, two students who contributed to this project. We've tried many different kinds of algorithms, including logistic regression, support vector machines, and random forests. And what becomes really interesting is that different algorithms can perform very differently in accuracy for the same species. For instance, SVMs are perfect in identifying this species, but the same species has a terrible accuracy with random forests. So that provides the challenge of, is there such a thing as an optimal algorithm to use for classification? Or does this depend upon the specific conditions that we're looking at? The other challenge is that classification accuracy drops with the number of classes. And obviously, there aren't only 20 mosquito species in the world. It's about more than 70 medically relevant ones. So as we expand our database, how can we keep our accuracy up? And these are challenges we're pursuing for the future. One important insight that we got from doing field outreach studies in Puerto Rico was the need for better noise cleaving. Here, we had two students, Sam and Jasmine, who looked at mosquito sound in red versus various noise models in blue and showed that simple principal component analysis can distinguish between mosquito sound and noise, which makes it very promising for us to build in automatic filters into our data collection pipeline. And of course, the holy grail question, can we use flight pattern variation as an infection marker? The system that we chose to study this was Aedes aegypti mosquitoes infected with dog heartworm. And this is because dog heartworm develops in the flight muscles of these insects. So if we see a signature in flight, it's going to be in this system. So we did this experiment where individual mosquitoes were recorded and then dissected to see whether or not they were infected how many larvae they had and in what stage. And some really exciting preliminary results show that infected mosquitoes have both a lower frequency and a lower propensity to fly, which gives us a lot of exciting possibilities for the future in pursuing this as an infection ID strategy. So this is to recap everything that I told you about what we did inside the lab. And of course, the proof of the pudding is in the eating out in the field, where we went ahead and did a bunch of recordings. We don't recommend that you catch the mosquitoes on yourself and get some Ziploc bags or something. But we did this cool field study with a bunch of friends in Big Basin Redwood State Park, just south of here, where we got a bunch of people, told them to hike along the trails and just record any mosquitoes that they found. And the map there, where every brown square represents a recording, shows that we can actually find spatial gradients even within this 1 mile by 1 mile region that we show here. In fact, when you hike along the trail right at the north up there, you can see that there's a hotspot of mosquitoes and very little up ahead, and that's because we were climbing uphill and the temperature suddenly got cooler and we started finding a lot more mosquitoes. So we can also see that the mosquitoes came out for dinner at around 7 p.m. that evening. And this gives us a spatiotemporal resolution that's far higher than anything that can be achieved by other methods of sampling mosquitoes in the field today. We also collected all the mosquitoes we recorded individually and bagged them, which gave us 80 tagged recordings to study variations out in the field. And this gave us insights such as size differences between wild mosquitoes do not actually translate into frequency differences, which is fantastic news because it means no matter where the mosquito came from, it's probably going to sound the same if it's of that species. There's another field trial we did in Madagascar. Out here, you can see Felix, myself, and Menza, our field tech out there. In Madagascar, we had two kinds of mosquitoes which obligingly sounded very different from each other, which was great. So we got a bunch of volunteers with phones, trained them for about 10 minutes, and told them to go hang out in the village one evening for 3 hours and record mosquitoes. So the squares again represent the recordings, and the numbers next to it represent the number of mosquitoes of those species from CDC light traps that were hung up in the same places over the same period of time as a reference standard for comparing how good our recording technique was. I'm happy to say that we collected more recordings than the CDC light traps collected live mosquitoes in each one of these cases. What's more interesting is that we can see a spatial species gradient where right up in the north, where there's a swampy riverland, you can see a lot of malaria-carrying Anopheles mosquitoes, whereas out in the hillside here, you don't see as many of them. And this is something that's reflected a lot more strongly through the recordings than from the numbers from the CDC lab traps. So then, after our field trials, where can we go next with this? It's said that when you make maps of mosquitoes, what you're actually making maps of is people who try to be interested in mosquitoes and record them. And what that means is that you need to get the 5 billion mobile phone users across the world to be interested in mosquitoes and start recording and tracking them. And this is what gave rise to a citizen science project that we call A Buzz, where we've roped in Rebecca, an artist, Christine, who's been doing a lot of web development for us, and Hazel, our lab manager, who's great on the outreach side. We've been inspired by a lot of platforms such as Eterna, which also grew out of a lab and became an interactive platform where citizens could engage in doing science, as well as iNaturalist, another ecology app that spun off from a master's thesis and allows people to collect observations of the natural world around them. Did you even know there was so much to see right in Palo Alto in our backyard? And the important thing about these platforms is the engagement with a citizen community out here, which is exactly what we're trying to build into A Buzz as well. We've prepared training materials such as this one that we can hand out to students, training videos that teach people how to record mosquitoes on our website up here. And I want to show you this cool video. So this video is from Sarah Zodi, our collaborator who did the work on the diarrhea, where she's teaching a workshop in Madagascar to school children on mosquito-borne disease and how to identify and record mosquitoes. So this is our ultimate goal through this initiative. This is where we've already got. We've started receiving data on our website, and here are some examples. This is from a high school student who was, out in Indonesia on a field trip. So it's pretty faint, but I hope you can hear the mosquito. And this one is from an elementary school student in Germany looking at invasive Aedes japonicus. So a 9-year-old did this, and that's amazing, right? This is the kind of data that we've also started receiving from other scientists. The Zodi lab at Auburn University sampled mosquitoes across every single county in Alabama that Sam did a lot of interesting work in processing. So to enable the collection of such data, we've started working on building our own app and web interface using this platform framework that's completely independent of whether you use an iPhone or Android or a computer. We've started making our screen designs, and we're getting into the exciting guts of how you actually code and test up an app and, check its functionality online. So you can see that we're actually able to record sound and edit files on our web interface. We have a 2-step rollout plan for this, where initially we want help from beta testers, entomologists who really know what species they're recording, to help us with unlocking new species on the tree of life of mosquitoes, and also recording baselines for new places on the world map. We're also going to reach out to programmers to help us develop better and better algorithms to classify our data, and finally go out into the phase of public rollout in a couple of years, where we can provide this as the home of all mosquito-related activity. So there's going to be coming soon. If a 9-year-old can record mosquitoes, so can up there on the top, as well as Abu Mayanak, who helped me figure out what a spectrogram was when I first started. Menza, who worked with us in Madagascar in setting up our field work and getting our field jobs up to scratch. Sarah, our collaborator from Auburn University, who did some amazing work that I've been showing you throughout, as well as Amalia and Saptarshi, who've been instrumental in helping us do field work and with conceptual contributions to the algorithms. I also want to call out Ellen from the CDC, who was incredibly supportive in helping us access a lot of mosquito species, and Malala, Matt, and Andres, without whom none of the work in Madagascar would have been possible. So with this, I want to take a little bit of time to thank some more people as well. I'd like to thank my committee for being here on this journey throughout with me. Firstly, Fabian, I'm really privileged to have been able to, run into you so often around the lab and hear your stories and your wisdom from time to time. Thank you so much for agreeing to be here, and I hope to keep chatting with you as long as I'm in the lab and even after. I also want to thank David. I've been told that this is his favorite portrait to use in talks, although the one that I really like is one where he has a model of, different woodcut things that show the progression of illness in malaria. So David was the one who first introduced me to the concept of dynamics and phase space and infectious disease, which is something I've got really hooked onto at this point. And it's been absolutely incredible, talking to you. I took my first real biology class with him as well. And, just talking about every topic under the sun, from dynamics and infection all the way down to how do you draw really good comic books and manga. I'm just so privileged to have interacted with you. I hope to keep this up forever. I also want to thank John. I hope there'll be no objections to keeping the acknowledgements in a lighter vein, because I've sworn not to be one of those girls who cries during their PhD defense. I thought that the Cafe was something special and, specific to Stanford. And then I went to Caltech and I heard that the best way to find John there was to basically go to the Red Door Cafe. But I want to say that, this spooky photo out here embodies something that I was absolutely blown away by when I first started interacting with him and he was in my committee. We had our first meeting and then the second meeting was like a few months later and I'd prepared a recap like, okay, I know that, this person would have forgotten everything I said. And I was amazed to find that every single thing that I had mentioned the last time was perfectly recollected and I just didn't have to recap at all. And I was completely amazed that, someone not only had such an incredible memory, but also took so much of an interest in my work and was willing to talk to me at that level. It was so cool to have interacted with you and got your really unique perspective on everything that I did so
It was
far.
the coffee.
Well, we got Beats coffee today, so I hope that was good enough. The other thing I really wanted to mention, for a long time, I thought science was just science, and it doesn't matter whether I have a male advisor or a female advisor to talk science to. And I thought this until I got Desiree and Erin on my committee and realized what kind of a difference that makes. So I put up this picture of Desiree here that I particularly like because of all the colors on her lab coat and how she brings so much color and fun and joy to science. Desiree has been so instrumental, especially in getting the Abbas work off the ground. It's like, without the kind of connections and support and input that you provided, we would not have got to half the position that we did. I also really loved that you taught me how to absolutely enjoy science and also really express myself, as a girl, as a woman, and just be the whole person I am as a scientist as well. Thank you so much for that. And I picked,
I'm wearing black.
I picked this picture for Erin because it is sort of like embodying the kind of exploratory, brave spirit that I've always associated with her. The very first time I actually saw Erin ever talk science was during her job talk, which left a very strong impression on me. And I was like, this was fantastic. I want to be here in a few years' time. And when Erin joined Stanford, I was really happy to get her on my committee. One of our first real conversations was something that absolutely blew me away with what she said. She was talking about her postdoc experience out in Utah, studying these wild grasses and how they were like no humans around for like miles and miles. And it was just her collecting all of this data. And I was like, wow, she is such a pioneer. And she's like, so bold and fearless, and I really want to do science like this and be, just as brave, I guess. So thank you so much for everything that we've talked about over these years. I've had so much fun with you too. You guys are like the best. Let's do karaoke again, please. I also want to give a shout out to Paul here, who is one of the most incredible teachers and mentors that I've had at Stanford. And I really want to say there are a couple of points, especially in the first few years, where I seriously considered dropping out of grad school. And at each of those points, Paul was the person who gave me the confidence in myself that, yes, I could stay on and make it all the way up to this day. Thank you, Paul, for everything. This is for everybody who thinks Manu walks in water. I do. And so do all these people standing on the bridge and watching him up there. This is from our field site in Madagascar, where people thought Manu was actually Jesus. Walking on the water, and he was going to like pan gold and make everybody rich in the village. I don't know how to capture, everything that I want to say about Manu in the short period of time, but this is what I think would encapsulate it best. I don't think I would have ever done research or done science or done a PhD had I not happened to stumble into Manu's lab 7 years ago. And 7 years down the line, today, knowing everything that I know now, in hindsight, there is not a single other place in the world where I would have not just done my PhD, but chosen to spend the last 7 years of my life. They also say Madagascar is full of endangered species. And another person I want to call out is the various shades of Felix here. So if imitation is the best form of flattery, then I think you should consider yourself extremely flattered. Like subtle ways and hopefully, like not too overt. I don't want to be a clone of you, but really following you around, working with you and interacting with you has just taught me so much about like how to carry myself as a person interacting in science. And I'm really, really glad to have had this postdoc who parachuted in and started working with me one fun day and just taught me so much about how to be a scientist and a person. So I'd love to call out the entire lab as it evolved over the years by name. I intended to write personal thank you cards to each one of you, but you gave me too much feedback on my slides yesterday, so I'll have to wait a few days for that. I want to thank my awesome family out here, in particular my granddad, who was, the biggest source of support for me and encouraged me every single time to get stuff done. My mom, my husband, Saptarshi, who many of you know and like. My aunt and as well as a shout out to my aunt, uncle, and cousins in Finland, and my cousins who've actually been awesome enough to make it here into the audience today as well. So with that, I want to leave you with a taste of the amazing things that you can do if you record mosquitoes. So hint, hint, go out there and do some more.
Thank you everyone for coming. You should enjoy this music for a little bit. This is a very special recording. We'll play this at the party that happens at the terrace, maybe in an hour or so. But now we'll have a closed door session with the committee members. So I'll request only the committee members to stay for. You can anticipate in the terrace for a little while.
So the public wants questions.
Do we have time?
Too late now.
How are you?
That was terrific.
Thank you. Thank you all for listening.